And our first speaker is Professor Alexandra Stolzing from Loughborough University in the UK. And she's going to talk about cryoprotectants, cryopreservations, and what this means for the field and how it can be used. So welcome, Alexandra. Thank you very much.
Well, thank you very much for the introduction. So that's me in the middle of the UK. That's where I'm located. And yeah, I will be talking about the rational design of cryoprotectants. Because one of the problems that we have in the field of cryopreservation is really that it's science that is not very advanced. Not a lot of people are using modern technology in this space. So we are desperately in need of people doing more. And I will give a few examples of what type of technologies you can use and what is currently missing. Just as a reminder for people that really work already in cryopreservation, it's the storage at ultra-low temperature, typically between 140 to 196, depending on your material. And it really can be everything. It can be biological samples. It can be your favorite antibody, your drug candidate. But there's also seeds, organs, and I will be today focusing on examples of the cell therapy field. So freezing cells causes cryodamage, mainly through and extracellular ice formation, but that's not the only mechanism. And that's already one area where we need more work. We really don't understand what is all going on, what happens with cells, how cells are reacting to this kind of damage. And to prevent ice formation, cryoprotectants are added to cells. But the problem is these cryoprotectants are really not perfect. So here are just a couple of examples of cell therapy products and where currently the survival rates are. So NK cell, CAR NK cells are really on the edge of translation. There are a couple of products really in development. And the survival is somewhere between 20 to 70%. So basically, you're losing 50% of the cells that you want to create. So it's a big problem in terms of manufacturing and cost. IPS survival has been worked on a little bit better. So we are at roughly 75%. HSC is something that has been in the clinic since 50, 60 years. We still only are at 40 to 60% survival. T cells, the same things. Sperms, same things. So we really kind of need to work on this. The CPAs that we're using are not fully effective. And they're also toxic to your cells. So here, just an example on IPS cells. So OCT4 or NANOC, if you take this as a marker of pluripotency, is reduced if you're using DMSO. You can have subtle changes in gene expression so that you have a drift within your cells in what they're doing. In terms of stem cells, you have changes in their preference to differentiate. You have epigenetic changes. So really a lot of things that can happen to your product and that might alter properties before they go into the patient. And the DMSO itself also is toxic to your recipient. So here are kind of just a list of publications talking about how toxic DMSO really is. It's causing simple things like vomiting, headache, but it could also lead to stroke, even to death. So this is a big problem. This is a survey looking at centers that are using DMSO in the cell product. And you see that the range is somewhere between 5% to 10% DMSO that is being used. There was one review in 2005 where they basically looked at this and said, okay, what's the side effects that we're observing and how many of our patients are observing side effects? And you see that about 50% of the patients have some mild to severe side effect. And then they recommended we really need to have a guideline. And then the same review basically was published in 2014. Same thing, we don't have a guideline. We don't really know what kind of level we should use. And 2020, same thing again. We don't have a guideline. So basically, it's kind of not regulated currently. You can do be very free in what kind of like CPA you use, and also how much DMSO you're using. But it's very clear that there are side effects to it. So there has been a lot of technology trying to either reduce the amount or wash the cells, remove it, or replace it. I will be talking today mostly about reduction of DMSO or a complete replacement. So to do that, we in my lab did the design of experiments, something that for the drug field is kind of really an old hat. So it's kind of just a way of thinking about like how you want to develop your product. So you look at like what factors you want to really measure. You're looking at like what levels you need. And basically you make long lists, and then you apply a statistical software to see kind of like what combinations and of factors in what concentrations you should be using. And it has no pre-concept on like, okay, I should have this combination, and the testing is kind of a better designed. There are many, many different factors that you can focus on, and this is just a list to give you like a snapshot of like what in terms of like factors you really need to think of. Are you using vitrification or cryopreservation? What temperature gradients will be using? Are you using an ice nucleator? What kind of volume are you freezing? Will you use computer-controlled freezing? And. So this is just kind of some of the factors that you could work on in improving. What we did is we went through the literature, and basically we looked at all of the longevity space. So our take was, what is good for like keeping an old cell healthy should also be maybe having some good effects in combating ice-induced damage. So that's what we did. We basically went through the literature, selected candidates, and that could include antifreeze proteins, that could include apoptosis inhibitors. So it was a pretty long list, and then we kind of like selected some candidates. In the end, we had like 24 of them. And then we did a toxicity range, because for some of these compounds, they have never been tested in cryo. So there was really no idea like in what range it might be effective on our cells. For cells, we use two types of cells, mesenchymal stem cells and endothelial cells. And then it could look like this. So there might be, these are very simple components. They are just kind of buffer ingredients. And you can see how different maybe your concentration range might look. So this was really necessary to narrow down which range would be kind of fine for our cells. And then next, we did the Plackman-Berman design. You can have up to 47 factor combinations, and what we were wanting to find out is kind of like what's the biggest positive influence on our cryopreservation. It's kind of a matrix, and basically the software spits out in what concentrations you should test in what order. And when you do testing, there are kind of like fine details. So this is just a slide showing you that classically, viability is often tested on day 1 after basically defrosting your cells. And that's the picture on the left. However, cells need a little bit of time for dying. So it's really bad to just look at cell survival on day 0, because you might be seeing really kind of nothing interesting happening. But then if you wait a little bit and you test it later, you suddenly see kind of like what a bad cryopreservation looks like. So there are kind of like caveats, and you need to think about this kind of stuff when you do your design. So don't do test cell survival on day 0. Here's a snapshot of kind of like being somewhere in the middle of this process. So there were in total 7 iterations of experiments that we did. And this just shows like some factor combinations that are good, some factor combinations that are completely disastrous, where you have basically no cells surviving. And yeah, we always optimized it for both cell types, because we were aiming for a solution that could be used kind of across different cell types. So next level, we do the two-level factorial designs. This time we're looking more like at interaction and like kind of like what works well, what works not so well. And so they are kind of examples. In blue is the things that really work well. Red is kind of telling you, now this is not a good combination. So again, like another round of iterations of experiments. And finally, we had candidates. And for the candidate validation, we also looked at differentiation for the mesenchymal stem cells, or we looked at like factor production for the endothelial cells because they were transduced with a factor produced to produce factor 8. So that's kind of like a functional test that your cells are not only alive, but they are actually functionally performing. And so this is data on the top for the metabolic, for the endothelial cells, and the bottom for the MSCs. We had always a solution that was commercially available with DMSO, 10%, or in terms of DMSO-free solution, another commercially available solution is kind of like benchmark. So whenever we were equally good as the DMSO-free solution, we were kind of happy. And as you see here, with our new two solutions, C and D, we were roughly in the same area. As I said, we also looked at differentiation. So for the MSCs, that was like bone or cartilage or adipose differentiation. And basically, we didn't see any significant differences. And then for the endothelial cells, we were looking at factor 8 secretion, and equally, they were performing as well as the commercial solutions. So we were pretty happy, and then also we looked a little bit more long-term to see, okay, now if the cells would be in your patient or your recipient, are they also performing long-term? Is there no subtle damage in the cells? And we could also observe after 8 days, and then we also did a two weeks check, and they were performing well. So proliferation was not damaged in any way. So that's a simple design, how that took roughly half a year, you can completely go free and design a solution from scratch. But we still have to do like the in vivo work for these combinations. But in general, this kind of like process was limited a little bit by us having to use vials. So it's really not very handy if you want to do kind of high throughput. And that's a problem in cryopreservation. So the second problem is that even if you have like loads of candidates and you think like, okay, I have a library of 10,000 factors, now let's do some proper work, not like, as I said, we had 20 combinations. The problem is high throughput is really not established. And I gave you some examples why that is the case. So first of all, we don't really have the tools. There are no plates available that you can seat your cells in that are stable at low temperature. Most of the time, you don't have like the right freezing equipment. You're kind of like putting it in a styropor box in -80. That's not appropriate. So one of the solutions here was a new type of device to cool plates. So here, the plates are magnetically linked to a block that is cooling down. So this time we had like a proper setup, at least for like a 96 well. But as I said, the plastic of the 96 well plates are actually not made to withstand such low temperature. And they warp. So basically, if you have ever seen it, I unfortunately never took a photo of it, but they really kind of mechanically get twisted. That means that your samples are unsterile, the cells will be mechanically stressed, but we did our very best to kind of like deal with it. And one other problem is that if you're using such plates, that the moment that ice is actually formed in your cryopreservation solution, it's completely random. So it can happen chaotically in your plate, and that's not really useful because you want to really tightly control this process. So one thing that you can do is you can do something that we call ice seeding. You basically put in a substance that causes to form ice in the solution. The problem is that all the stuff that was available wasn't suitable for a cell therapy product that would go into patients later. So we looked at something that could be autoclaved, that is kind of inert, and that is not in touch with the cells. And that's kind of the design on the bottom. Basically, we had an insert that contained the iStart that was in the solution, but was sealed off. So this little foot that you see there, this is where at the bottom, there was the ice-seeding substance kind of was encased there. So it was in contact with the liquid, but not in contact with the cells. If you want to know what it is, it's basically what creates rain in your clouds. It's a little bit of like feldspar. So it's kind of like a very inert product. Usually what people are using is bacterial proteins, not very nice for your cell therapy product. So if you look at cooling now, we had a little bit more even cooling. But so this is kind of like showing this process. So basically, this is just showing the plate, and that we had very inhomogeneous kind of like survival and temperature curves. So on the right side, you see all the curves in orange are the ones that are kind of like with the ice starter, and the ice formation is at a slightly higher temperature, and that's good because that means there is less damage to your cells. And also another thing that is happening is that there is something that we call like an edge effect. So kind of samples might freeze more easily at the edge of your plate versus the middle. So that's unfortunately also still a problem because you have more plastic at the edge. So there is like uneven like heat transfer happening. Another thing that needs to be worked on. So now if we do this comparison and we look at the viability of the cells, with iStart, we really had much better results than without. So that's something that is slightly better. I would say it's still things that we have to work on. So ice nucleation kind of is getting better, but the design of the insert is still not perfect. And also it's only available currently in a 96 well setup. So again, like if you're thinking about like scanning a library that has 100,000 compounds, that's not very useful. Next bit is a storage solution. As I said, a lot of people are storing their plates, if they work in plates, at -80. Also not great, so we actually need kind of like towers that are holding plates, something that is currently not available. As I said, also suitable plasticware is not available. The plastic that it's made of is not mechanically stable at low temperature. So in summary, I think there's a lot of room for improvement. And a lot of this could come from actually the longevity field, because as I said, we're dealing more or less with the same cell stresses, like there's some damage, some oxidative stress, some apoptosis induction. If you have a compound that mitigates this kind of damage, it might also be useful in a cryopreservation compound. We need more systematic approaches. I also would love to do like a machine learning on like gene expression data from cryopreserved cells. There's barely any of this data out there, so it's actually nothing there to like train your model on. So that's definitely a problem. High throughput approaches are difficult because you don't have the tool. And yeah, the last is thanks for the people that sponsored this research, and also the people that helped me work. The ice seeder and the new cryopreservation tool were actually from a company called Asymptote that were now acquired by GE Healthcare, and they are very innovative in the space, but we need more of them. Thank you.
Thank you, Alex. Indeed, very cool. Like new technologies, probably like 15 years ago, that's where like longevity was. So we need like people who come in and develop those technologies. Does anybody have any questions as of now? We have time for one or two. I have a question. If you think about it, it's like that a lot of things that work in cell lines or in worms or in mice don't work in humans, because you just have like different metabolism that we don't fully understand yet. But do all those cells freeze similarly, or do we have to figure out cell-specific freezing, or once we figure out cryopreservation and those cryoprotectants, that it's easier to transfer between like different systems?
No, unfortunately, every cell line needs to be worked on, and we have no idea why they react so differently. That's why I'm saying like we really need more like basic understanding, basic cryobiology data. There are some cell lines that are super tough, but they are like the NK cells are very, very fragile. And so we currently don't really understand why. If we would have that knowledge, then yes, a more rational design could be applied.
So if you would say, what are like your top 2 most important things people should work to make the next steps?
Gene expressions and tools.
Cool. Okay, then thank you
very
very much, Alex.